over to Stefano, then Frits Sobels Award winner, EMGS 2020. Amazing work, Stefano.
So I must confess that all these persons talking about me was really, I'm really impressed. And okay, forget it. And I will start to say thank you very much. And I will say thank you very much many times during my presentation, because of course I have to say thank you very much to so many persons that came with me along this journey which is about 30 years long. And therefore there are so many persons that were important for me as a colleague, as a scientist, as a friend also. And this is the reason for the title, actually. This is a journey done by an epidemiologist that started the kind of environment in which epidemiology was sort of a foreign science. I mean, nobody, when I started, everything was has to do with genetics. I mean, and people in the lab organized their work. And so I really started this collaboration, and that was very exciting because there was so much to do, so much to explore. And however, before I really started this travel together, and I have, okay, no, I don't. I can use this. Aiuto. Okay, here we are. And I want just to spend 1 second, 10 seconds to mention Frits Sobels, because I'm sure that there is many people in the audience that they don't know anything about Frits. Actually, I never had the opportunity to meet him, because when he retired, I just started, so there was not this opportunity. However, what I want to say is there was a special person, because he was an outstanding scientist, but he was also an outstanding organizer. And I really need in our activities this kind of persons which are doing science, but also they spend their time in organizing, working in societies, and making serving young investigator, serving increasing the possibility of networking in the society. And this is exactly what he did. I mean, starting big activities like mutation research journals that are leading journals in the field, or starting new societies, or organizing many more activities. So it's definitely an example for all of us, science, but also dedication to people in work, doing this, especially to young investigators, but everybody working in the field. Why I say, I forgot which one was this one. Okay, and why I said about this is a journey, and because it is I mean, in the last 30 years, there has been a lot of changes. I mean, can you imagine, when I started in the 90s, there was still the idea of one gene, one protein issue model. And now we are dealing with genomics, metabolomics, proteome, all the omics science. And so there has been in this last 3 decades, so many changes. And I've been so lucky to be part of this process. And so I really wanted to go to spend this presentation with you, just to go through what happened in this period and mixing what has been the main achievement of the career and the most recent advances. Because of course, I'm very happy to take the opportunity of this presentation to share with you new results. Okay, this is to maintain your attention, of course. And well, what you see on the top corner is the first paper I've published with biomarkers. I mean, when I was working in the occupational environment, when I was working in the National Cancer Institute in Genoa, there was the group by Angelo Bondandolo. They asked me to make some statistics about the study on pesticide workers, and we did it. At that time, the role of epidemiologist was just to make students test chi-square test and maybe ANOVA. And so we published the paper, but I really realized that there was a number of knowledge gaps. And so at the end of the paper, I just tried to list these gaps. And these gaps, this black hole of knowledge, gaps of knowledge, are something that guided my career. And actually, there was the very serious problems in the study design, in the statistical analysis of data. Not to talk about exposure assessment, of course, just pesticides was pesticides. You can imagine how many metabolic compounds are in the pesticides. Then biomarkers. At that time, we knew very little about the biomarkers that we were using, the variability, the role of host factors, the main confounders. So we didn't know a lot. And especially the last point was, okay, you can observe DNA damage, genomic instability in some subjects, but what is the result of this damage? Can we expect to observe long-term effect? So that was these are a number of challenges, and these challenges are what I've done during my career, essentially, at least until now. And so I wanted just to go through the first part, which is dealing what's about study design and statistical analysis. However, I forgot to say one thing, that in this period, I collaborated a lot with people that are, I mean, I'm now receiving the Frits Sobels Award, but there are two other Italian scientists that received the award before, and there are Eugenia Dogliotti and Silvio De Flora, and they have been really source of inspiration for me, and their career has been really critical. So thanks to them for their work and for their friendship. But back to statistical analysis, the real challenge was that, okay, we have biomonitoring studies that were human studies, but they kept using the genetics approach as a geneticists. And then the very first challenge was to push everybody working in human biomonitoring studies to use epidemiological methods, to realize that even before molecular epidemiology, of course, and so realize that this is a real epidemiological studies, and so we have to use these proper methods. And just to make the statistics, if you look, I put here a few papers that we published on this topic. In the very first paper that was published in 1994, you see that what was impressive was that only 5% of population studies were analyzed using multiple regression analysis, multivariate analysis. That means that 19 out of 20 studies used just chi-square, students t-test, univariate analysis. So there was a lot of space for improvement. Improvement that lasted a lot, and now we are just making new advances. I wanted just to share with you some, just to name them, because if you have to analyze data on using count biomarkers. When I say count biomarkers, I say biomarkers like micronuclei, chromosomal aberrations, that you score maybe 10 out of 1,000 or 10 out of 100. So this is really count. And so just if you have to analyze this kind of data, take into consideration the possibility to use methods like take into consideration extra Poisson variation. I mean, these are now quite standard in advanced statistics, in which you can use not only the variability of the outcome, but the variability of all covariates, and to see all of them how to contribute to the overall variance. And so to model in the best way, I mean, your data. The same like, you know, this kind of count biomarkers quite often have a lot of zeros. I mean, we are dealing with rare events, because when you have like 2, 3 events per 1,000, there's a lot of cells with 0. So there is a variability of zero, the deep models, how they are called, and in which you can properly shape the variance of the model. And finally, that's very commonly used, the multi-level count modeling, because we are more and more working on pooled analysis, in which data from several countries, several labs are put together. But this kind of data are generally have a hierarchical structure. I mean, there is the country, there is the lab, there is the operator, there is the tissue, and so on. And so we can really shape this variability. Okay. And this is kind. I want to say one thing. I have done these studies and this modeling with Marcello. Marcello Ceppi has been working with me for 30 years. He's not the kind of person working that you can see in the meeting, but he's an outstanding statistician, and we have published together best paper in the field. Now something is more. I told you this is a journey which is a continuing journey, a journey that takes new path, and a new pathway is really towards clinics. I mean, the idea that more and more people are using this kind of biomarkers in clinical studies, just to anticipate the diagnosis, or to predict the response, or to trace this as a potential therapeutic target. And therefore, has it happened when we had to move epidemiological methods in genetics, now we have to use clinical epidemiology methods. And so this is exactly what we are doing. And I'll make you an example of recent results. The first, the use of meta-analysis. I mean, I saw that generally people making review, they use narrative review, more recently systematic review. But actually, the meta-analysis is a quantitative estimate of effect, which comes from clinics, in which generally is used to estimate the effect of a drug. So now, if you want to use this in molecular epi and in human biomonitoring studies, we have in summarizing data from human biomonitoring studies, we have to use these quantitative methods. Just to show you one paper, for instance, we have just submitted together with Randa Elzain, which is #1 in genomic instability in respiratory disease, but also with Maxim Asinov, as a postdoc Russian that is one of the persons that survived working with me in Rome. You see here is a typical forest plot in which we can reanalyze. You can laugh, but it helps. And it makes a quantitative estimate that we can really use. But there is another approach that I want to share with you, that when you publish a randomized clinical trial, you have to use what is it called a minimum information criteria, because people, before accepting your study, has to know exactly everything about your study. So, for instance, the consort criteria is a criteria that say how many people you included in the study, how was the randomization, how many people left the study, why left the study, and so on. Or the PRISMA for the meta-analysis, or the STROBE for the observational epidemiology. We never did this in molecular epi or in our studies. So together with people from the H-Comet project, and I will talk about later about this bunch of people, of the wonderful people, and I say we put together, especially with the work of Peter Möller and Sabine Langi, but also with Amaya, with Roger, and the other, the minimum information criteria for the Comet essay, MIRCA. So the paper is in second revision, and I hope that will be accepted. And then the idea is that when you publish a study using the comment essay, you have to make everybody aware of the methods that has been used and what has been done. So this is a paper that is possibly published quite soon. Now, let's go on, let's continue the journey. And another step, another important stop by was with the validation of biomarkers. And this is really critical because we didn't know, we know so little about biomarkers that we are using. So the natural background of this biomarker, the frequency of this biomarker that you can expect, or what are the confounders that most predict the biomarker, that most affect biomarker, or what is the role of host factor, what is effect modifiers, and so on. And so this is what I've done for several years. And I started with, actually I started with the Scandinavian, the collaboration with Scandinavian countries. I say something more about them. But I saw that Jenny Dogliotti joined the mess. Ciao, Jenny. Glad to see you. And I want to say that I've done a lot with Scandinavian scientists. I say more about them, but the real work was done with the human project that started at the end of in 1997. It was the ICM in Toulouse, and the idea is that I mean, I will just skip this and I show you this nice slide I've received this morning from Michael and from the other people in the committee. And I want to really thank all of them. I mean, you see people, Errol, Nina, of course, Hannu, Claudia, Sigi, and but I especially want to thank Mike Fennell. Thank you for joining me in this journey. We have spent a lot. I saw that Claudia joined the meeting now, and thank you, Claudia, also for you are in the perfect moment for joining. And for that was a huge effort. You see from here that a lot of that's the very first international project that we lead together, and it started in 1997, involving several thousand subjects. And I say we had a lot of time. Also, we had also a lot of fun. And I don't know if I touched something. Okay, you see that the secret of these groups is that not only to have science, but also to have our time to create a network, not only scientific, but also human network. With this group, we published some papers that were very critical in assessing the effect of age, gender, assessing the role of micronuclei, in this case, as a biomarker of aging. And I would like to spend just a few minutes more, a few seconds more about one result that was published in like in the 2003. Thanks to Monica. Monica that heavily contributed. Thank you, Monica, for surviving me for so many years that we spent together. And Monica was very precious for me. And you see the results here. These are the results from the studies that were involved in the human studies concerning smoking. Smoking, yes or not? And that was quite puzzling. That was unclear why smoking, which is the most important carcinogen, did not move actually micronuclei frequency. And the results are occasionally positive, occasionally negative. But the power of thousands and thousands of subjects allow us to put together data by number of cigarettes smoked. And so we were able to find out this kind of shape in which it was quite clear that smoking increases genotoxic damage and genomic instability. But it needed, but the sensitivity of the assay was not the best. And so we needed heavier exposure. So that was quite critical, because when people has to design a study on micronuclei, for sure they have to take into consideration the number of cigarettes smoked. And then another example from a very recent study here is the moment to say thank you very much to Mirta, not only for presenting my candidature, not always because it's being the mother of Isabella, the wonderful Isabella, but especially because you have been working with me more than 1 year in Rome, and several years in assembling 20,000 subjects. I mean, we were able in this year, within the framework of the HCOMET project, to put together 20,000 data from over 50 laboratories. And one of the results is, for instance, is here. You see here the expected most frequent frequency of different endpoint of the comet assay, the tail length, tail intensity, tail moment. So if you plan a study using these endpoints, you can expect to have a result in this range. So this is a very important guide for new laboratories, especially. The paper has been submitted and still we are waiting. But now I want to spend the last 10, 12 minutes I have in the most important issue. That is, we have a biomarker which is positive for chromosomal aberration. Is we have a higher level of damage. But what's the final effect? And we started this study with the Italian and Scandinavian collaboration, because and the idea was to validate that I mean the causal pathway is from an exposure and a favorable environment through intermediate endpoints, then to cancer or chronic diseases. We cannot measure that. We measure just surrogate events like biomarker, like micronuclei, chromosomal aberrations. The idea, can we validate the idea that people with a high level of these biomarkers have also a higher risk of cancer, of chronic diseases? This is what we did in between the two centuries, in the century, at the end and the beginning of the century, with the help and with the leading of Lars Hagmar, especially. I want to thank Lars, and a special memory for him, because unfortunately he passed away too young, maybe 10 years ago. He was a very good friend of mine. I still hate him because he's dead of 10 years and his age index is much better than mine. So this is just something to say that this was really a good one, and I miss him not every day, but really often. And the results were published at the end in 1998, but in 2008 we were able to put together a new group linked in Scandinavian, Central European, and Italian data. We were able to put together 22,000 subjects from 11 countries. And the result of this cohort study assess clearly that subjects, when they are healthy, but they have in the top tertile of the distribution of chromosomal aberration, has a higher risk of developing a cancer, of die of cancer. And this data was replied a few years after with the micronuclei. And you see here the group is smaller, but the contribution is very international. And you see that Michael told me that this paper that we published in 2007 has been cited over 1,000 times now. So it was very important in the field, assessing micronuclei as a risk of biomarker. And now I want to say thank you very much to my most recent colleagues and friends. I mean, people in the HCOMET group. I mean, I would like to start from Andrew and Maria, that we started the idea together, but actually they were the driving force. And in the group, then I met a number of outstanding scientists, young scientists. So I was so happy to be there, to spend time with them. So JP in particular, my brother JP, Sabine, Gudrun, Mirta, Amaya, I mean Roger, Susanna, Luna, all the other. So thank you very much for your friendship and for this collaboration. You see here that also the H-Comet SA created a very large network of laboratories, 50 laboratories, 20,000 people. And we were eventually able to create a small cohort, and the results, which are now in preparation, but we plan to submit it in September after vacation, show quite clearly that, not so clearly as a chromosomal aberration, but clearly enough, you see highly significant, that subject with the highest level of, when they are healthy, subject with the highest level of DNA damage has a 38%, a significantly higher risk of die. And if we restrict the analysis to remove accidental mortality, this association is even stronger. And you see here also the survival curve showing quite clearly as people with the highest level has a much worse probability of surviving than people with low level of DNA damage. And that was mostly what they have done, but something there is more. So what next? I mean, the journey is going on, and what is really changing the landscape here is that medicine, clinical research has realized that biomarkers, early effect biomarkers can be important, can be predictive, can anticipate diagnosis, can predict progression, can be a therapeutic target. And on the other way, people working with biomarker has realized that our biomarkers can be useful for clinical studies. And furthermore, we have realized that biomarkers can be a lot. I mean, we can have a very huge number of different biomarkers that can be used to understand what's going on, to understand the intermediate processes of several causal pathways. And then this is the magic of personalized medicine. You see here that the personalized medicine is in the same way is putting together environmental, personal, economic, social determinant that interact together with host factor, gender, age, gene expression or gene modulation, and the biological expression of chronic disease. That is the first step leading to the clinical feature of chronic disease. All of these provide an individual feature which is the basic of personalized medicine. And we can say a lot with our biomarkers. And then I wanted to make a very few couple of minutes to say, to show an example of how these kind of biomarkers can be used for personalized medicine, which is the case of frailty. I mean, I've been using this with several other fields. I mean, like COPD, with the help of Patrizia Russo, working a lot with me in this last period. We are working in this biomarker in psychiatric or in geriatrics. But what I like a lot is really frailty, because frailty is really important, because changes is a predictor of disability, or is in older ages, in veterans, in old persons, is predictive of disability, mortality, health cost, hospitalization, and is associated with several events. So the diagnosis is based on clinical features. So the hand strength, if you lose weight, the way you jump or you climb a stair. I mean, we can say more. We can increase. We can help the diagnosis of this condition using our biomarkers. And this biomarker has been developed thanks to the collaboration with the Coruña group. I mean, I would like to thank Vanessa. Vanessa is another survivor. She spent 2 years in Rome, and that was difficult for her. Because I'm not really a good organized person. I may be very nice, very friendly, but when we talk about work, I'm not so well organized. So people working with me has to keep really the right direction. And Vanessa did it, and so did Maria when she comes, and thanks also especially to Blanca Lafont for this wonderful collaboration. And we published data on the micronuclei that was really important. And using the cytome assay, we realized that we can have an increases in this condition associated with micronuclei and lymphocytes, binucleated cells, and a reduction, a mirrored by a reduction by condensed chromatin or karyolytic cells. And the next slide is about the use of, we use also telomeres and other biomarkers, and immunological markers. I mean, I know that also Michael is submitting papers on immunology, and more we are creating a large landscape of biomarkers that can be, that each biomarkers can contribute a little piece of information. So we need to collect all piece of information and to create better possibility for the patients or for the subjects that we can anticipate their disease. They can treat them, or you can treat them even before they get healed. And the very final slide, I want to stay in the minute, otherwise Sabine tells me that I'm a long talker, is I mean, I found this when I decided to call this to make a journey in the title. I found at the end this very nice slide, which is actually a never-ending journey. You see a corner, you don't see what is behind the corner. And I want to end my presentation just saying thank you very much to a couple of not person, because I want to say thank you to my institution, that gave me the possibility to come here in Rome. Thank you to the San Rafael University and the Research Institute for that. And especially thank you very much to my wife, Angela, and to my family for joining me and making possible this journey. So thank you very much for your attention.
Really, just massive congratulations, thoroughly deserved for this Frits Sobels Award, and it's amazing to see your career, and I hope you continue to support the EMGS and inspire everyone to work even to some proportion of your expertise and output throughout their career. So congratulations again, amazing achievement. Maybe claps from people. If there's nothing else, then I think we can sign off as an excellent meeting, and we will see you next time.